I believe almost of everyone in the hall has experience on taking care on patient wound. So, have you ever encountered patient with unhealed wound or chronic wound? I believe most of you would say yes. Today, I am honored to present our innovative wound dressing with therapeutic potential. Globally, 8.2 million people have been affected by chronic wound, which this leads to severe health complications or deaths. While antibiotic and antiseptic exist, they are slow and have side effects. Due to this, we concerned and we sought to find alternatives and found andrographolide paniculata, or in Thai, Fatalai, known for its anti-inflammation and anti-infective property. However, its poor solubility presents a challenge, leading us to develop an innovative synthesis. Our hydrogel dressing, which were made from polyvinyl alcohol and sodium alginate containing bFGF, will undergo releasing tests combined with the preparation of andronanoparticle, which will examine cytotoxicity tests. For the objective, first, it is to examine the hydraulic property of bFGF hydrogel and safety of andronanoparticle. Second, to evaluate the efficacy of andronanoparticle bFGF hydrogel on wound healing and antibacterial in rat skin. Third, to determine underlying mechanism regulating wound healing in rat skin. And for the hypothesis, bFGF hydrogel had high releasing and andronanoparticle were safe assuming cell viability. And this enhanced wound healing effect in wound healing and reduces bacterial colony with thermal growth factor response. So, starting with the evaluation of hydraulic property of bFGF, which the concentration will be focusing on the testing of the sodium alginate concentration, which you can see in, it shows in 25%, 50, 75, and 100%, which the optimal release efficacy was observed at 25%. In the time-dependent manner, and in addition, cytotoxicity tests confirmed that andronanoparticles are safe at concentration below 0.04 microgram per microliter. And to avoid menstruation or Astra cycle effect, vascularity changes, and for the wound healing, Melissa red were used in this study. In which they will then subject to acclimatize and subject to anesthesia and make surgical wound with the treatment for 21 days. And please note that the orange refers to the control group, while the green represents the andro bFGF group. And this color pattern will be consistently used across all slides. Moving forward to the wound contraction rate, image of wound healing over time demonstrated that andro bFGF hydrogel significantly improved bone contraction in day 7 and day 14, highlighting its therapeutic potential, whereas for the bacterial ACA sample from day 14, it shows numerous colony in the control group compared to the andro bFGF group, which significantly reduces colony in the andro bFGF group, confirming its antibacterial efficacy. Next, this slide shows changes of histological morphology stained by H&E, which this image shows pattern of macrophage and fibroblast in 4 time points. The circular refers to the pattern of macrophage, whereas the spindle shape refers to the fibroblast, which we had prepared the figure showing its trends. By the x-axis refers to days, and for the y-axis, it refers to the number, which we'll be focusing on the lower phase first. The purple line refers to the macrophage in control group, while for the red one, refers to the andro bFGF group. And it can be noted that it shows significant in day 3 during the inflammatory phase. And for the upper part, it refers to the fibroblast, which the brown line is the control, whereas the blue line refers to andro bFGF group. And it does show significant increase in day 7 and day 14 because of proliferation phase, which triggers collagen synthesis, confirming that our andro bFGF hydrogel has the property of decreased inflammation and help recruiting fibroblasts. Furthermore, mesothelioma shows changes in histological morphology stained by the mesothelioma, which this shows percentage as we focus on the collagen deposits, particularly in the gray color, and significant change were observed on day 7 and day 14 due to the reflecting of collagen recruitment during the proliferative phase, and day 14 showing the remodeling phase. And this image shows mRNA expression pattern of dermal wound healing marker, interleukin 6 and bFGF were significant on day 3 and day 7. And for the TGF-beta, it do show significant in day 3 and day 14. And for the VEGF, it does show significant in day 3, 14, and 21. Interestingly, VEGF had shown an obvious result changes when compared to the other genes, so we would like to study in further protein levels. For the VEGF protein level expression, it was significantly higher in the andro group compared to the control group on day 21, indicating that it enhanced angiogenesis during the late stage of wound healing. These results do align with mRNA finding, confirming that our andrographolide extract upregulated both VEGF genes and protein levels. According to the finding, the andro-bFGF microsphere hydrogel accelerates wound healing and promotes wound closures and reduces bacterial colony. It synergistically enhances mediator recruitment during the inflammatory phase, stimulation fibroblast proliferation, and support collagen deposit during the remodeling phase. In conclusion, all mentioned results will help improving the beneficial use of our hydrogel formulation, enhance wound healing effect, and anti-infection, along with highly dermal growth factor response. These transcending all boundaries of medical research and prepare wound dressing alternative for global health. In further study, 3D bioprinted skin model or rabbit can be used to stitch skin irritation, followed by clinical trials are going to be done in healthy human, and when it passes, chronic wound human representative will be chosen on the study and then launched to the market. These are all our references and our acknowledgement. Thank you for your attention.
Please accept our thanks for the great presentations. Do judges have any comments or any questions?
Did you compare the hydrogel with the standard treatment or not?
Yeah, we have compared. So we have the control group, the andro-bFGF. As we have prepared the group. Our control is the PVA/SA hydrogel. And for the standard treatment, we didn't compare, but we do have the control as I've mentioned.
And what kind of bacteria that you use for your experiment?
Okay, so those bacteria will be found on rat skin, which it can show up like the Staph aureus, which is can be found on the rat skin. And for the normal flora, it can show the Staph epidermidis, which can also be found on the rat skin. Yeah.
How many times did you perform your experiment? Do you repeat the experiment or just one time?
Yeah. We have repeated the experiment for 3 times. Yeah. Okay.
All the results is the same, consistent?
Yeah, it shows the consistent result, and these are all presented on the slide. Yeah.
Could you elaborate more about that you mentioned that you have performed experiment 3 times, and how many animals each time, and how many animals in total?
Okay, so we have prepared the experiment, which we have the sample of 12 rats, male vista rat, yeah. And we divided into like parts, like we focus on different time points, so we didn't focus, but we have 4 times for the 5 times for the experiment. I mean the time point.
So you mean you have 5 time points and 6 animals each? So you have 30 in total?
I want to clarify. We have 12 animals in which in day 0 we doesn't kill the rat, and in day 3 we have we focus on 0, 3, 7, 14, and 21, right? And in day 3 we just start the experiment, and in day 0 we start the experiment, and in day 3 3 rats were killed, in order to focus. And then for day 7 also repeated process for triad, and day 14 also, and day 21.